Why would an ion move across the cell membrane at all? Why doesn't it just stay where it is? The answer lies in the difference, or gradients, in ion concentration and electrical charge across the cell membrane. Imagine dropping a drop of dye into a container of water. The dye immediately spreads throughout the water, away from where it was originally concentrated. This process is known as diffusion. Diffusion can be thought of as a chemical force driving ions, or any molecule, from where it is in higher concentration to lower concentration. This is called moving down the concentration gradient. There will be a force toward movement down the concentration gradient until concentrations are equalized. Consider this example, showing a larger concentration of sodium ions outside the cell. Without an open sodium channel, the ions can't cross the cell membrane and have to stay where they are. If a sodium channel in the membrane is open, sodium ions will flow into the cell until concentrations on the two sides become equal. Ions are also subject to electrical forces. When positive and negative charges are separated, for example, on opposite sides of the cell membrane, this creates a voltage difference between the two locations. We can measure these voltage differences using an electrode, often just an insulated metal wire with an exposed tip, or sometimes a thin glass tube filled with salt water. This electrode is attached to a voltmeter or oscilloscope, which will compare the voltage at two locations. Imagine the way that the positive ends of two magnets will push against each other. Similarly, one positive charge will experience a force pushing it away from many other positive charges. As a result, there will be an electrical force on ions pushing them to move so that voltage differences between areas are eliminated. So positively charged ions, like sodium or potassium, will experience an electrical force driving them to flow from higher to lower voltage, bringing the voltage difference between those two locations closer to zero. By convention, when measuring the voltage difference across a membrane, we consider more positive charges inside the neuron to be a positive voltage difference across the membrane. So in this example, there's a voltage difference across the membrane of about -70 millivolts, which means that a positive ion like sodium will experience an electrical force to move from outside to inside the cell. So if a sodium channel is open, sodium ions will flow down the electrical gradient. There will be an inward electrical force on the sodium ions until the voltage difference across the membrane reaches zero. Whether ions of a particular type will flow in or out of a neuron can be determined by a combination of electrical and chemical forces that those ions are experiencing. Sometimes both forces push ions in the same direction, as in the case where sodium ions are in greater concentration outside the neuron and the voltage difference across the membrane is negative. Sodium ions moving from outside to inside the neuron are moving down both the electrical and concentration gradients. If sodium channels in the membrane open, sodium ions will flow into the cell. Other times, the electrical and chemical forces of ions of a particular type are in opposite directions. If one force is stronger, ions will flow in that direction until the strength of the two forces is balanced. At that point, there will be no net flow of ions across the membrane. The voltage difference across the membrane when that state of equilibrium is reached is called the equilibrium potential for that ion. For example, here we see potassium ions more concentrated inside the cell, with a negative voltage across the membrane. In this case, the electrical force on the potassium ions is inward, but the diffusive force is outward. If the concentration gradient is large enough to overcome the electrical force, some potassium ions will flow out, making the voltage across the membrane even more negative and the electrical force even stronger. As this process continues, eventually the two forces will become equal and net flow of ions across the membrane will cease. You can calculate where this balance will be reached using something called the Nernst equation. The exact value will depend on the concentration gradient across the membrane, the charge of the ion, and also the temperature. You can perform this calculation separately for each type of ion. In the next video, we'll talk about how these forces, along with the opening and closing of ion channels, create action potentials, the basis for electrical signaling in neurons.